morning, everyone. We are from Science Society.
Good morning.
So today we are going to share interesting content on two topics, neuroscience and virus in a brand new way.
Yes, and let's start the video. Hope you all will enjoy it.
If I were to ask what is the most important organ in your body, what will be your answer? The heart, the lungs, or the liver? I'm sure most of you will answer the brain, which gives us high intelligence and control of our body. And it's exactly for this reason that our understanding of this mysterious organ is quite limited compared to its vast potential. There is much more going on in our heads than we think, no matter how empty you think it is. First, let's start with some fun facts. Think carefully, judging from your experience, can you control your heart rhythm, sweating, or the motion of your digestive organs? Not really. This is because they are all involuntary actions controlled by the autonomic nervous system. What you can voluntarily control is limited to the somatic nervous systems, which is in charge of mainly your skeletal muscles, so that you can move your arms, hand, legs and body. Diving deeper into the topic, we can find two sets of pre-installed undeletable programs that activates automatically to respond to different situations unconsciously. The sympathetic nervous system will be in charge of the fight or flight response, while the parasympathetic nervous system will carry out the rest and digest response. Imagine yourself in a critical situation like performing on the stage or seeing something you are scared of. You will experience a behavior where you find yourself sweating and your heart racing. This is what happens when your sympathetic nervous system gets activated and takes the lead. It is used for encountering dangers and ensuring survival. On the contrary, the parasympathetic nervous system will be active at times where you feel the most relaxed, like during sleep. It is mainly used for conserving energy for growth and metabolism. That's why we know that physical health and mental health are interrelated. In some cases, people who suffer from anxiety and great stress have prolonged increase in heart rate and blood pressure, stomachache, excessive sweating, etc. And it is due to the constant activation of that fight or flight response. So remember to take regular rests to relax your mind, or you might explode. Let's get back into topic. How does the nervous system give commands to the whole body? This important task is completed by a huge number of neurons. In fact, there are 86 billion neurons in our brain. But how exactly are nerve impulses transmitted within and across neurons? It involves two separate types of messages, electrical and chemical. We'll be talking about the electrical part in this video. The basic mechanic is just similar to conducting electricity. Normally, the transmission speed is very slow at about 2 meters per second with neurons alone. Therefore, most of the neurons in your brain looks like this. The blue regions are myelin sheath, just like the plastic wrapping on copper wires. You can imagine the blue region as a highway, where the transmission of nerve impulses there will be accelerated to nearly 200 meters per second. And the gaps between two myelin sheaths are like gas stations where the nerve impulses are amplified to prepare for the next propulsion. At this point, some of you might ask, how do we know all these information? Are they mere speculation? In recent decades, we are able to obtain more evidences not only from microscope observations and dissection, but also from electroencephalogram and functional MRI. They are essential tools for us to gain a deeper understanding on the brain. How does it work? For electroencephalogram, it detects the electrical activity of the brain when neurons fire and represents the data as waves on a diagram. You might have heard the word MRI. What's the difference between functional MRI and the standard one? Actually, they are quite similar, but the main difference would be that fMRI detects the brain activities at particular regions, while MRI images the physical structures of body parts. Therefore, fMRI is especially useful for finding out the active portion of brain when a person has a particular action, thought, or speech. Now that we have covered the basics, let's move on to deeper and more interesting topics. Try recalling the last time when you remembered having a memorable dream. What happened during it? Did it make any sense at all? What people usually feel is that the scenes in dreams feels indescribably familiar to some experiences in awake time, but the scenes just doesn't make any sense. For example, try identifying some items in this room. I'll give you some time to work it out with your friends. At first they may all seem familiar, but most of you might not even know what the brown thing in the front is. This image is actually generated by an AI with the objective to make unrecognizable things, while making them feel familiar. Dreams work in a similar way, feeling real yet not. Why is that so? In fact, we do not actually see or hear during the day. The feeling of seeing or hearing something is not objectively existing. It is the brain taking in information of light, sound waves, etc., producing a sensation, and combining these sensations into a complete experience in the sensory area. In dreams, however, the brain takes in little information. Instead, the brain itself activates the sensory area, thus producing a similar real experience. At this point, what if I tell you there's a process that you consciously know you are dreaming and can even control how the dream progresses? These dreams are called lucid dreams. Lucid dreams are not out of your control like watching a movie, but controllable as if you are the director. While scientists are still unsure why this happens, it is known to be a consciousness process. Even though dreams often feel familiar, but why do we often cannot recall what happened during it? And why it feels as if time has warped since you fell asleep to the moment you wake up? The secret lies in the memory forming process. When information first arrives at the brain, it records them as sensory memory and then short-term memory. But one of the drawbacks is that our short-term memory lasts for an incredibly short time, anywhere from 5 seconds to a minute. To form memory, a key step involved is the conversion of short-term memory into long-term ones by the hippocampus. It is like adding a word to the dictionary. Next time when something similar happens that triggers your memory, it is like typing that first letter to an autocorrect system and then the whole word will appear. However, information received during sleep are not added to the long-term memory. So when you wake up, the memory is already lost and we feel like the last moment we experienced is ourselves lying on bed. Forgetting things that happened during sleep is no big deal, but imagine forgetting important events in your life on a regular basis. How does it feel? This is exactly what dementia patients experience. Normally, when we are not retrieving the information for a long period of time, memory is lost because the neuron connections become weaker. But those with dementia might experience memory loss much more frequently and quickly. Particularly for Alzheimer's disease, more of a certain kind of protein is present in patients' brains. They are believed to affect neuron connections and functioning, just like blocking some circuits in a computer. That's why those with Alzheimer's disease might experience loss of memory and impaired thinking. These findings show that neurological functioning is related to physical changes in the brain. In the past, we often think that the brains are very flexible to changes in children and babies, but not in grown-ups. This turns out to be wrong, as scientists found that neurons and new connections are constantly formed, and they give such phenomenon a fancy name, neuroplasticity. Therefore, next time when someone tell you that motor skills like learning an instrument or sports can only be trained at a young age, and it is too late for you to learn a new skill, take it with a grain of salt. For the musicians out there, it is even proved by some studies that perfect pitch can be developed by repetitive, dedicated practice. In fact, neuroplasticity also applies to studying and reciting. By spaced repetition and active recall of facts, you are constantly strengthening the connections between neurons. This helps you remember the facts in textbooks. This chart is widely known as the forgetting curve, that illustrates you will remember nearly nothing if you only listen in lessons or read textbooks, but not consolidating knowledge or being repeatedly exposed to the same content. Back to dreams. Why is the flow of time in dreams different from the real world? Imagine being trapped in a black room with no natural light and any indicator of time, like clocks and watches. How can you know the time of day? The human body is designed to be in harmony with time. Biologically, our body has the circadian rhythm, also known as the biological clock. With inputs from your eyes, your brain constantly adjusts the rhythm. When the sun is blazing, it is calibrated to noon. When the sun starts to fade into nighttime, your brain receives this information and produces more of a kind of chemical messenger, making you feel tired and go to bed. But what if there is no natural light to calibrate our circadian rhythm? In a research, some volunteers are invited to live in a dark cave for a few months. During this period, they reported that their conscious mind cannot notice the difference. Every sleep and wake cycle feels as normal as the 24-hour one. However, in fact their circadian rhythm is disrupted to become a 48-hour cycle. They work for 36 hours straight, then sleep for 12 hours. Why is it 48 hours? We don't know, just as the answer to many other questions in this field. Let's move on to the funnest part today, in my opinion, so you get to try these illusions and have fun tricking your brain. Here you can see that the circle is moving according to the arrow inside it. It even changes shape according to the arrows. However, you can see here that the circles aren't actually moving. Visual illusions are quite fascinating. The things our eyes see aren't always accurate, as the brain can interpret the data it receives incorrectly, which causes illusions. Moving on, it's time for some myth busting. Myth 1, you only use 10% of the brain. This is a very common myth. It is actually false. You actually use almost all parts of your brain at any given moment, even during sleep. All parts of the brain are involved in specialized functions for your daily life. So, myth busted. Myth 2, male brains are more logical, female brains are more empathetic. The second one is a bit stereotyped, that male brains are more logical while female brains are more empathetic. This is once again false, and whether one is more thoughtful or empathetic depends more on individual variations rather than biological basis. Sometimes, researches like these might be biased due to social expectations and gender stereotypes, and are not entirely trustable. So, myth busted. Reaching the last part of our presentation, we also want to share about some interesting technologies in the neuroscience field. One of the major directions is to help the disabled to regain their movement/senses. Biotechnology companies like Neuralink successfully help those who lost their arms or are paralyzed to regain fine control of their body, like precise movements of fingers, by implanting very tiny chips into the brain. Bionic eyes work quite similarly, also involving the implantation of tiny chips, and it helps blind patients to regain sight. But don't expect to be able to shoot lasers from your eyes like Superman, while still being able to see in the near future. Furthermore, neuroscience advancements brings us the chance of prevention and early intervention of neurological disorders like Alzheimer's disease. It is lately found that blood tests can be used to predict patients' risk of developing various conditions by detecting the certain protein we have mentioned before, so we can give treatments before that protein starts to excessively build up in the brain. Lastly though, some scientists are even more ambitious to map the brain activities and their corresponding meaning, potentially making mind reading possible. They utilize functional MRI to estimate neurons' responses in different activities, so as to correspond and make a dictionary-like archive.
So, which part of the neuroscience sounds the most interesting to you? Personally, the technology part got me the most. Think about the applications of chip implantation and brain mapping. They can incredibly help on our way to unleash the vast potential of the human brain.
Now, it might be exciting to think about, but is such technology truly beneficial to human beings? I mean, at least before clear directions are made to clarify the ethical dilemmas and safety issues it brings.
Well, I agree with the safety part. The human body and especially the brain is very sophisticated. We know what might happen if something goes wrong. It is possible for brain tissue to get damaged during the surgery or by the chips. Furthermore, the idea is still very new, and any long-term adverse impact is unknown.
I understand the concern of you guys. There's a great difference between using an implantable brain chip on curing disorders versus enhancing the human brain. It's fair to argue that brain enhancement is unethical, but isn't it counterintuitive to delay the development of potentially life-changing or even life-saving technologies just because of these concerns?
Yes, of course, I agree that they can help patients, but in my opinion, as long as chip implantation thing is used ethically and safely, we shouldn't worry about it. The problem is that we cannot completely avoid the possibility of malicious intentions by some people. Inevitably, pioneers in this field are trying to create chips that connect to computers. This is like a deal with the devil, which comes with great cost. What could possibly be worse than a hacker gaining control of the chip in your brain?
This is a great point. Moreover, I want to add that this also intensifies inequality if implantation is allowed. The development of brain chips is monopolized by some companies, and it is no surprise that they must commercialize and monetize their products. Then, only the very rich can afford to enhance their abilities like memory and information retrieval.
Fine, let's move to brain mapping. It just makes use of MRI and is much less intrusive than implanting brain chips, but the result is promising. There's once a case in which scientists are able to communicate with vegetative state patients by detecting brain activities. The patient can answer simple yes or no questions by imagining a scene. At last, the patient was asked if he wanted to remain in such a state or cease any life-supporting medical procedures.
That's a sad story. But I do agree that brain mapping can have some medical applications. For example, it can help stroke patients who cannot speak or move to communicate with the world outside via thinking only. It might be indirect, but this might improve their quality of life.
I would not exclude the possibility of mind reading helping patients, but I'm quite concerned about it. Imagine each of your thoughts can be detected and interpreted by AI and seen by others. There is no privacy at all, even in thinking.
After all, each new technology comes with a bright and a dark side. It all depends on who make use of it. It certainly would be better if the general public can set some red lines and regulate those who might violate the common ethics.
Okay, so let's hear what our audiences think. On the screen now, you can find 2 inquiry questions for you. 1, evaluate the pros and cons of implanting chips into your brain. 2, if you are given the chance in the future, would you want to get a chip implanted in your brain? Why or why not? You can capture this page and discuss with your peers later.
So it's about time we should start the second part of the video. What is about it again?
Oh, why are you always forgetting things? Did you get brain fog? Anyways, let's dive into the video about virus.
When you hear the word virus, what's the first thing that pops into your mind? It must probably be COVID-19, which has been wandering around the entire globe for the past few years. But how do they actually work? They are non-cellular particles that causes diseases. Examples include the familiar COVID-19, which is caused by a kind of virus called SARS coronavirus 2. They disrupt cellular mechanisms in order to replicate themselves and infect more cells. Furthermore, much more diseases like influenza and the common cold are caused by viruses too. Your cell is just like a food factory. Each of them has their own cookbook that stores information of how to keep the factory running. Normally, cookbooks are photocopied into recipes and given to chefs in the factory to produce food. Imagine some random armed people hijack the factory and give chefs recipes of poison. Eventually, both the armed people and the chefs died from poisoning, and the factory closed down. Now in biological terms, DNA are like an archive of cookbooks, and RNA resembles individual food recipes. When the virus enters the cell, it is hijacked to produce virus particles from viral RNA. Finally, the cell dies and the viruses produced leave the cell. The viruses infect other cells, so on and so forth. Hey, but are we defenseless against viruses? No. Never underestimate our bodies. We have the immune system to help defend against invasion of viruses. Now some of you might be like, yes, it is common sense that we have white blood cells. Well, hold on, your body has different types of guards like B cells, T cells, phagocytes, the list goes on. They patrol the body and kill any viruses on sight. Our immune system is amazingly sophisticated, but sometimes it just isn't strong enough. So centuries before, we invented vaccines to bring our immune system on steroid. You might have heard about this story, but we are going to tell it anyways. In 1786, Edward Jenner discovered that if one person had caught cowpox before, they wouldn't catch smallpox. Then, a scientist's curiosity drives him to do something weird. He scratched cowpox pus into a child's skin. Later, the child is exposed to smallpox virus, but he didn't catch it. This is the origin of vaccination. Diving into its mechanism, let's use another analogy. Initially, cowpox viruses enter the body, and they are like criminals who are smart enough to all use the same identity card. The guards take some time to recognize them, but eventually they are all arrested, and the guards remembers their identity card. Next time, smallpox viruses disguise into normal citizens, but soon they are recognized and arrested because they hold a similar identity card as cowpox viruses. Eventually, most people are vaccinated, making it harder for smallpox to enter cells and infect people. So nowadays, smallpox is eradicated completely from the world. It is a huge triumph for humans in the campaign with smallpox, but not for other viruses. Pathetically, smallpox is the only virus humans have managed to beat. As we all know, our campaign with COVID is a failure. Why is that so? The answer lies in the structure of smallpox virus. They contain DNA inside, which is like 2 long magnet chains sticking together. It is hard for you to remove one specific magnet and insert another one. In other words, DNA is very stable, and its structure rarely change. So smallpox virus never mutates in its entire history into different variations. In contrast, other viruses such as COVID-19 and influenza contains RNA as its genetic material. RNA is like a single magnet chain. You can easily replace an individual magnet with a different one. In other words, RNA is relatively unstable and prone to change. Therefore, COVID-19 mutate more quickly into a whole lot of variations, from alpha to beta, then Omicron BA point something, the list goes on. Think mutation as random balance changes to a game. Viruses can mutate to give different stats ranging from infectious to non-infectious, and not fatal to very fatal. As a result, some become very weak, but some become overpowered to dominate. The virus variant that is more infectious but less fatal will become the main strain under natural selection. That's why Omicron became most prominent for a whole year. Unfortunately, we still have a long way to go in fighting this pandemic. Therefore, to buy time for research and development of vaccines and medicine, the strategy of isolating and curing infected patients is employed to control the pandemic at its initial stage. The first step to achieving such strategy is screening out possible patients. However, distinguishing COVID-19 from influenza is impossible just by observation, as their symptoms are nearly the same. While both influenza and COVID-19 are similarly infectious, the original strain of COVID-19 has a death rate of 1% compared to 0.001% of influenza. Therefore, without effective measures to identify and cure patients of COVID-19, a lot of fatalities might be caused. How can we prevent this from happening? Initially, viral RNA test is used to identify infected citizens. It works by searching for presence of viral RNA in samples of nasal fluid and saliva. If the RNA of a specific virus like COVID-19 are present in the samples taken from the upper respiratory tract, we know that viruses are replicating there. In other words, the person is infected. To make viral RNA tests more accurate, polymerase chain reaction is adopted. It works like a printer, photocopying the genetic material of the virus. One copy becomes two become four, and vice versa. We call one cycle of PCR as one photocopying procedure. After the whole process, billions of copies of genetic material can be found, so it is much easier for detection. During the pandemic, you might have heard the word CT value in press conferences by the government. Simply explained, CT value refers to the number of PCR cycles required for successful detection of viral genetic material. Imagine that you have $1 and one of your classmates has $10. For each cycle, your wealth doubles. Who will be able to afford a car worth $1 million first? The answer is clear, the one with $10 initially. So, if the concentration of viral RNA in the sample is higher initially, less cycles will be required for the amount of genetic material to reach the common threshold. Therefore, a lower CT value means a greater amount of virus in a person's body. The PCR test is accurate, but very time-consuming, and requires help from the professionals. What if we want a quick test of COVID-19? I am sure all of you are familiar with it, the rapid antigen test that you do every day. But how does it work, and why stuff like orange juice or cola give a fake positive result? To answer the first question, we must understand antigen and antibody first. The red spike protein here acts as antigen, and antigens come in different shapes. An antibody is a structure like this. If the shape are compatible, antigen and antibody combine. So, if the nasal fluid sample contains virus, then there will be antigen, and vice versa. Then, let's watch a video on this topic. In short, with antigen in samples, the flowing antibodies will deposit on the T line, giving color and indicating a positive result. However, even if there is no antigen in samples, flowing antibodies will still deposit on the C line, so there is always at least one line. Recently, a drug against COVID-19 is developed and under limited prescription. They work by stopping the virus from replicating their genetic material and producing more virus. Hence, severe damage to body cells can be prevented. Up till this moment, we are depicting viruses as evil organisms, but can viruses, by any chance, be helpful in certain occasions? It is rare, but there is. Ever heard of bacteriophages? Bacteriophages can infect bacteria and kill them. In the Ganges River located in India, there's a lot of them to keep the dirty water a bit cleaner. This explains its self-purifying property, which is often portrayed as holy water in superstition. However, similar to human beings, bacteria have their own defense mechanisms to fight against their rivals. The CRISPR gene in bacteria was discovered recently. It works like a scissors to freely cut RNA or DNA. When bacteriophages try to inject RNA into bacteria, the RNA is cut into pieces, so they become non-functional. The talented scientists apply this scissors to gene editing. It is no exaggeration to say that the CRISPR gene is another revolutionary discovery of our times. When used in gene therapy, many diseases like cancer can be cured.
Wow, the CRISPR gene seems really powerful. In the future, we can apply this into medicine, farming, and many more fields. What do you guys think?
Personally, I'm quite excited with it. CRISPR can be useful for gene editing, especially in the medical field. Apart from targeted therapy, we can have another groundbreaking new treatment for cancer, the T-cell therapy. It makes use of Cis9 protein to modify immune cells, allowing them to target cancer cells and kill them. I can't wait to see the applications on treating genetic disease too.
Well, it's good that we are one step further on the campaign with cancer too, but I have mixed opinion on the latter one. CRISPR gave us the ease in genetic engineering, but ethical and safety discussion cannot be overlooked. Say, for example, this technology is relatively new, but it involves changing of DNA. We wouldn't know its long-term effect for now. Maybe it might mess with the proto-oncogene or tumor suppressor gene, then backfiring us by inducing cancer instead.
It sounds logical to me that we couldn't evaluate its long-term health impacts for now. Moreover, treating genetic disease by altering the DNA of the fetus comes with so much more controversy. Even if it comes with a good intention of curing people, genetic engineering of embryonic DNA will alter each and every cell of the fetus, and even its descendants. This severely undermines the need of informed consent in medical treatment, and both the fetus and its descendants cannot talk and decide their fate.
I do agree with both of you here, and I think the law has proven justice in this case. Previously, a group of scientists used this technique to alter DNA of an embryo, making it less susceptible to HIV. Later, they are sentenced to imprisonment and huge fines. I believe this gave a clear message to the science field that we must not sacrifice the ethical standards.
That's great, but still, technically, it's hard to define whether a trait is a genetic disorder or not. And so ambiguity in law might happen. For example, if you regard mental retardation as a disorder and change it, then those with normal IQ would be comparatively the new group of intellectually disabled.
I think what you want to say is human enhancement using genetic engineering. As much as I emphasize the inequality problems arising from chips implantation, the same goes for gene editing too.
Anyways, let's also see what our audience think. There's two inquiry questions too. One, evaluate the possible benefits and drawbacks of genetic engineering. Second, do you think modifying human gene is morally acceptable? Remember that when you approach questions in the science field, especially biology, do you think about ethical arguments too, other than pros and cons?
I think that sums up our discussion today, and it's about time we have to end. If you, fellow audiences, want to learn more about the human immune system, you can look for the book Immune, A Journey to the Mysterious System that keeps you alive from libraries or bookstores.
Also, don't forget that there are also online resources out there that make learning much more interesting. You can find animated videos with captivating illustrations on YouTube like these channels.
By the way, if you want to learn more about neuroscience, viruses, and other topics about science, why not join the Science Society and discuss with our members?
Yes, and you can also expect interesting activities in Science Society because our theme next year is Butterfly Conservation Extinct at Munsang.
We integrate butterflies, plants, and adrenal automation in the butterfly greenhouse. Hope that we can see you in our meetings next year.
Bye.
Bye.
Bye.